Welcome back to my channel LinkedIn Farmer. Today's video is based on the fundamental unit of life, chapter 5, class 9. Before starting this video, please like and subscribe to my channel LinkedIn Farmer. Let's start. Introduction. The fundamental unit of life is the cell. Cells are the smallest structural and functional entities in living organisms, serving as the basic building blocks of all living things. They carry out essential processes such as metabolism, growth, reproduction, and response to stimuli. Cells can vary in size, shape, and function, but they all share common features, including a cell membrane that separates the internal environment from the external surroundings. The study of cells, known as cell biology, is crucial to understanding the intricate workings of life and the complexities of living organisms. The exploration of cells began in the 17th century with the invention of the microscope, allowing scientists like Robert Hooke to observe and describe cells in cork. However, it wasn't until the 19th century that advancements in microscopy and scientific techniques led to the realization that cells are the basic units of life. Matthias Schleiden and Theodor Schwann formulated the cell theory in the mid-1800s, stating that all living organisms are composed of cells and that cells are the fundamental units of life. This breakthrough laid the foundation for modern biology and our understanding of the intricate workings of organisms at the cellular level. What are living organisms made up of? Living organisms are made up of cells. Cells are the fundamental structural and functional units of life. These cells come in various types and perform diverse functions within an organism. In turn, cells are composed of molecules, including proteins, nucleic acids, DNA and RNA, lipids, carbohydrates, and other organic and inorganic compounds. These molecules work together in complex networks to carry out the processes necessary for life, such as metabolism, growth, reproduction, and response to the environment. The intricate interactions between cells and their components give rise to the remarkable diversity and complexity seen in living organisms. Certainly, here are some point-wise activities that can be observed using a compound microscope when examining an onion bulb. Preparation of the onion thin section. Peel a thin, transparent layer from an onion bulb. Place the thin section on a microscope slide. Staining the cells, optional. Apply a drop of iodine solution to the onion section on the slide. Iodine can help enhance the visibility of cell structures by staining certain components. Placing the slide on the microscope, gently place a cover slip over the onion section to prevent air bubbles. Adjusting the microscope, place the slide on the stage of the compound microscope. Use the cos adjustment knob to lower the objective lens close to the slide without touching it. Look through the eyepiece and use the cos adjustment knob to bring the image into focus. Observing different cell structures, observe the thin layer of onion cells under low magnification, 10x or 20x, to get a general view of the cells. Switch to higher magnifications, 40x or 100x, to examine cell structures in more detail. Identifying cell components, observe the rectangular or polygonal shape of onion cells. Notice the cell walls, which appear as dark lines surrounding each cell. Identify the central vacuole as a large, clear space in the center of the cell. Observe the nucleus as a darker, rounder oval structure within the cell. Examining the nucleus, focus on a single cell's nucleus using higher magnification. Observe the distinct boundary of the nucleus. Note any visible nucleoli within the nucleus. Observing cytoplasm and cell wall. Focus on the cytoplasm surrounding the nucleus. Observe any granular structures or organelles within the cytoplasm. Note the difference in staining intensity between the nucleus and the cytoplasm. Observe the cell wall surrounding the cell, outside of the cell membrane. Recording observations. Draw labeled sketches of the observed cell structures, including the cell wall, cell membrane, nucleus, and cytoplasm. Experimenting with light intensity, adjust the diaphragm or light intensity to observe how it affects the visibility of cell structures. Comparing cells, compare the structures of different onion cells within the thin section. Taking photos, optional. If a camera or digital microscope is available, capture images of the observed cell structures for future reference or analysis. Remember to handle the microscope and slides carefully to avoid damage and to achieve accurate observations. Cell and its discovery. The cell is the fundamental unit of life, serving as the basic building block of all living organisms. The discovery of cells and the formulation of the cell theory marked a pivotal moment in the history of biology. Here's an explanation of the cell and the story of its discovery. Cell definition. A cell is the smallest structural and functional unit of living organisms. It is enclosed by a cell membrane and contains genetic material, organelles, and other components necessary for life processes. Discovery of cells. Early observations. The exploration of cells began in the 17th century with the invention of the microscope. In 1665, English scientist Robert Hooke examined a thin slice of cork under a simple microscope and observed small, box-like structures that reminded him of the cells, small rooms, in a monastery. He called these structures cells, a term that persists in cell biology terminology to this day. Anton van Leeuwenhoek, Dutch scientist Anton van Leeuwenhoek, in the late 17th century, further advanced the field by developing more powerful microscopes. He made detailed observations of various biological samples, including water droplets and animalcules, microorganisms, which expanded the understanding of microscopic life. Cell theory formulation. Matthias Schleiden, 1838, a German botanist, Schleiden, proposed that plants are composed of cells and that the cell is the basic unit of plant structure. Theodor Schwann, 1839, a German physiologist, Schwann, extended Schleiden's ideas to animals, suggesting that animals are also made up of cells. Rudolf Virchow, 1855, a German physician, Virchow, added the concept of cell division, proposing that all cells arise from pre-existing cells. Cell theory established mid-19th century. The combined work of Schleiden, Schwann, and Virchow led to the formalization of the cell theory, which states, all living organisms are composed of one or more cells. The cell is the basic unit of structure, function, and organization in all organisms. All cells come from pre-existing cells through cell division. Significance of the discovery. The formulation of the cell theory revolutionized biology, providing a unifying concept that explained the organization and functioning of all living organisms. It paved the way for understanding the complexity of life processes, including reproduction, growth, metabolism, and heredity. The development of microscopy techniques further advanced our knowledge of cells, revealing intricate cellular structures and organelles that are responsible for specific functions within cells. In summary, the cell, as the smallest unit of life, has been discovered and explored through the collaborative efforts of various scientists over centuries. The cell theory, which emerged from these discoveries, forms the cornerstone of modern biology, enabling us to comprehend the remarkable diversity and complexity of life on Earth. The microscope's ability to reveal this hidden microcosm revolutionized our understanding of life, leading to profound advancements in fields ranging from medicine to genetics. The discovery of cells not only established a unifying principle for biology but also underscored the remarkable complexity and unity shared by all living organisms. Living organisms can be categorized into two main groups based on the number of cells they are composed of. 1. Single-celled, unicellular organisms and 2. Multicellular organisms. Single-celled organisms. Single-celled organisms consist of only one cell that performs all necessary functions of life. These organisms are complete entities on their own. Examples of single-celled organisms include 1. Bacteria. These microorganisms are unicellular and come in a wide variety of shapes and forms. They play crucial roles in various ecological and biological processes. 2. Protists. Protists are a diverse group of eukaryotic microorganisms. Examples include amoebas, paramecia, and algae. 3. Yeast. Yeasts are single-celled fungi that can carry out fermentation and are often used in baking and brewing. Multicellular organisms. Multicellular organisms are made up of multiple specialized cells that work together to perform specific functions. These organisms exhibit a higher level of organization and complexity. Examples of multicellular organisms include 1. Humans. Humans are complex multicellular organisms composed of trillions of specialized cells organized into tissues, organs, and systems. Different cell types perform various functions, such as nerve cells transmitting signals or muscle cells facilitating movement. 2. Plants. Plants are also multicellular organisms. They have various cell types, including root cells that absorb water, stem cells that provide structural support, and leaf cells that carry out photosynthesis. 3. Animals. Animals, ranging from insects to mammals, are multicellular organisms with distinct cell types. For instance, nerve cells allow for communication, while blood cells transport oxygen. In summary, single-celled organisms consist of a solitary cell that carries out all life processes, while multicellular organisms are made up of multiple specialized cells that collaborate to perform complex functions essential for survival. Activity of single-celled organisms. Single-celled organisms exhibit remarkable adaptability in diverse activities despite their simplicity. For example, bacterial reproduction. Bacteria, being single-celled organisms, reproduce through a process called binary fission. This involves the cell dividing into two identical daughter cells, each capable of independent existence. This rapid reproduction contributes to the rapid growth of bacterial populations. Amoeboid movement. Amoebas, a type of single-celled protist, showcase a unique mode of movement known as amoeboid movement. They extend their pseudopodia, temporary protrusions of the cell, to push their cell body forward, allowing them to change position and capture food. Yeast fermentation. Yeast cells, being single-cell fungi, perform fermentation to convert sugars into alcohol and carbon dioxide. This process is utilized in baking to make bread rise and in brewing to produce alcoholic beverages. Activity of multicellular organisms. Multicellular organisms exhibit complex behaviors and activities that arise from the collaboration of specialized cells. For example, human muscle contraction. In humans, muscle contraction is a coordinated activity involving different types of muscle cells. Nerve cells transmit signals to muscle cells, leading to their contraction. This intricate interplay of cells allows for movement and locomotion. Plant photosynthesis. In plants, leaf cells work together to perform photosynthesis. Chloroplasts within these cells capture sunlight and convert it into energy, which is then used to synthesize glucose from carbon dioxide and water. Ant colony behavior. Ants, as part of a multicellular society, exhibit highly organized behaviors. Different types of ant cells fulfill various roles within the colony, such as worker ants gathering food, soldier ants defending the nest, and the queen laying eggs. In essence, single-celled organisms engage in activities that enable them to survive and reproduce as individual units, while multicellular organisms showcase complex behaviors that emerge from the coordinated interactions of specialized cells, contributing to the overall functioning and survival of the organism and its social group if applicable. What is a cell made up of? What is the structural organization of a cell? A cell is made up of various components, including genetic material, DNA or RNA, a nucleus, in eukaryotic cells, cytoplasm, organelles, such as mitochondria, endoplasmic reticulum, and Golgi apparatus, and a cell membrane. The structural organization of a cell includes several components. At its core is the cell's genetic material, either DNA or RNA, housed within the nucleus. Surrounding the nucleus is the cytoplasm, containing organelles such as mitochondria, endoplasmic reticulum, Golgi apparatus, and more. These organelles perform specific functions like energy production, protein synthesis, and cellular transport. The cell is enclosed by a cell membrane, regulating the passage of substances in and out. This intricate organization enables cells to carry out essential life processes. Plasma membrane or cell membrane. Definition: the plasma membrane, also known as the cell membrane, is a thin, semi-permeable barrier that encloses the contents of a cell. It separates the cell's internal environment from the external surroundings. Composition: the membrane is primarily composed of a lipid bilayer, which consists of phospholipids arranged with their hydrophilic, water-attracting, heads facing outward and their hydrophobic, water-repelling, tails facing inward. Functions. The plasma membrane serves various crucial functions. 1. Selective permeability. It controls the movement of substances into and out of the cell, allowing only specific molecules to pass through. 2. Protection. It provides a protective barrier that shields the cell from harmful external factors. 3. Cell signaling. Membrane proteins play a role in receiving and transmitting signals from the environment or other cells. 4. Cell adhesion. It facilitates cell-to-cell adhesion and interactions. 5. Transport. Membrane proteins assist in active and passive transport of ions, nutrients, and waste products. 6. Endocytosis and exocytosis. The membrane is involved in the processes of engulfing materials into the cell, endocytosis, and expelling materials from the cell, exocytosis. Membrane proteins. Integral proteins are embedded within the lipid bilayer, while peripheral proteins are attached to the membrane's surface. These proteins serve various roles, including transport, signal reception, enzymatic activity, and structural support. Cholesterol. Cholesterol molecules are present within the lipid bilayer. They help maintain membrane fluidity and stability. Fluid mosaic model. The arrangement of lipids, proteins, and other molecules in the membrane resembles a mosaic pattern, where the components can move laterally within the lipid bilayer. This fluidity allows for flexibility and adaptability. Cell recognition. Carbohydrate chains attached to proteins, glycoproteins, and lipids, glycolipids, on the extracellular surface contribute to cell recognition and communication. Eukaryotic vs. Prokaryotic. While both eukaryotic and prokaryotic cells have cell membranes, eukaryotic cells also have internal membrane-bound organelles like the nucleus, which are further enclosed by membranes. Dynamic nature. The composition and properties of the plasma membrane can change in response to various factors such as temperature, fur, and the cell's physiological state. In summary, the plasma membrane is a vital structure that defines the boundary of a cell, enabling it to interact with its environment while maintaining its internal conditions. Osmosis activity within egg. Materials needed: raw egg, clear glass or container, vinegar, water, measuring cup, marker or tape, paper towels optional. Procedure. Preparation. A. Place the raw egg in a container or glass. B. Carefully pour enough vinegar into the container to completely submerge the egg. The eggshell will dissolve in the vinegar due to its acetic acid content. Observation phase. A. Allow the egg to soak in the vinegar for 24 to 48 hours. You'll notice bubbles forming around the egg as the shell dissolves. The egg's outer membrane should remain intact. B. Gently remove the egg from the vinegar using a spoon or tongs and place it on a plate or paper towels. It will be soft and rubbery due to the shell being dissolved. Osmosis demonstration. A. Carefully observe and handle the egg. You'll notice that it has increased in size due to the process of osmosis. This is because water from the egg's surroundings, vinegar, has moved through the egg's semi-permeable membrane to balance the concentration of substances inside and outside the egg. Hypertonic solution, water bath. A. Fill a clear glass with water. B. Measure the egg's diameter and mark it on the glass using a marker or tape. This will help you track any changes in size. Placing the egg in water. A. Gently place the egg in the glass of water. B. Allow the egg to sit in the water for several hours or overnight. Observation phase continued. A. Check the egg's size after it has been in the water for a while. B. You should observe that the egg has become larger compared to its original size in the vinegar. This is because water molecules from the water have moved through the egg's membrane to areas of lower solute concentration within the egg. Explanation. During the initial phase of the activity, the eggshell dissolves in the vinegar, leaving the egg's outer membrane intact. This membrane is semi-permeable, allowing water to move in and out. When the egg is placed in water, osmosis occurs. Water moves from the area of lower solute concentration outside the egg to the area of higher solute concentration inside the egg, causing the egg to expand. This activity demonstrates the principles of osmosis, where water molecules move across a semipermeable membrane to balance concentrations on both sides. It's a hands-on way to observe osmosis in action and understand its role in maintaining the water balance of cells. Cell wall. Definition: the cell wall is a rigid and protective layer located outside the cell membrane in plant cells, bacteria, fungi, and some other organisms. It provides structural support and helps maintain the shape of the cell. Composition: the cell wall is primarily composed of cellulose in plant cells. In bacteria, it consists of peptidoglycan, while in fungi, it contains chitin. These components form a tough and durable framework. Rigidity in support: the cell wall's rigid structure provides mechanical support, preventing the cell from collapsing under pressure or changes in osmotic conditions. It contributes to the overall shape of the cell and helps maintain the integrity of tissues and organisms. Protection: the cell wall acts as a barrier that protects the cell from physical damage, pathogens, and environmental stressor. Permeability: the cell wall is porous and allows certain molecules to pass through, but it is generally less permeable than the cell membrane. Its selective permeability helps regulate the movement of substances in and out of the cell. Growth and development: the cell wall plays a significant role in cell growth and expansion. As plant cells grow, new cell wall material is deposited, contributing to the cell's size increase. Cell communication: plasmodesmata, small channels that traverse the cell wall, allow communication and transport of materials between adjacent plant cells. Lack in animal cells: animal cells lack a cell wall. Instead, they are surrounded by a flexible cell membrane that provides structure and protection while allowing for movement. Bacterial cell wall types: bacterial cell walls can be classified into gram-positive and gram-negative based on their response to the gram staining technique. Gram-positive bacteria have a thick peptidoglycan layer, while gram-negative bacteria have a thinner peptidoglycan layer surrounded by an outer membrane. Fungal cell wall: fungal cell walls contain chitin, a complex carbohydrate similar to cellulose but with different properties. Chitin provides strength and rigidity to fungal cells. Evolutionary significance: the presence and composition of the cell wall have taxonomic significance and can help classify organisms into different groups. Cultural and practical uses: plant cell walls are a source of dietary fiber in human nutrition and are used in various industries, including paper production and biofuel generation. In summary, the cell wall is a crucial structural component that surrounds certain cells, providing support, protection, and a degree of regulation in terms of permeability. Its composition varies among different organisms and has diverse functions in maintaining cell integrity and facilitating growth. Cell wall activity: observing osmosis in Eloda cells. Materials needed: fresh Eloda, waterweed, stems or leaves, microscope slides and cover slips, microscope, water, salt solution, diluted, dropper or pipette, microscope or hand lens, optional. Procedure. Preparation. A. Place a few Eloda stems or leaves in a container of water to keep them fresh. B. Prepare a salt solution by diluting salt in water. The solution should be hypertonic to the Eloda cells, meaning it has a higher solute concentration than the cells. Mounting the Eloda. A. Place a drop of water on a microscope slide. B. Gently remove a small section of Eloda leaf or stem and place it in the water drop on the slide. C. Add a cover slip over the specimen, making sure to avoid air bubbles. Initial observation. A. Observe the Eloda cells under low power magnification. Focus on the cell walls, cell membranes, and any chloroplasts within the cells. B. Note the shape and appearance of the cells. Adding salt solution. A. Use a dropper or pipette to carefully add a small drop of the salt solution to the edge of the cover slip. Allow the solution to slowly spread under the cover slip. Observing osmosis. A. Observe the Eloda cells under the microscope after a few minutes. Focus on any changes in the cell's appearance. B. You should observe that the cells have become slightly plasmalized. The cytoplasm has pulled away from the cell wall due to the loss of water. Comparison and conclusion. A. Compare the plasmalized cells to the initial appearance of the cells in water. B. Discuss the role of the cell wall in preventing excessive water loss and maintaining cell shape under hypertonic conditions. Explanation. In this activity, you observe the effects of osmosis on eluda cells when placed in a hypertonic salt solution. Eluda cells are surrounded by a cell wall, which provides structural support and prevents the cells from bursting. When placed in a hypertonic solution, water molecules move out of the cells, causing them to lose water and become plasmalized. The cell membrane detaches from the cell wall, highlighting the role of the cell wall in maintaining cell shape and preventing excessive water loss. This activity demonstrates how the cell wall functions in conjunction with osmosis to regulate water movement and maintain cell integrity. It also provides a hands-on opportunity to observe the effects of different solute concentrations on plant cells under a microscope. Nucleus. Definition. The nucleus is a membrane-bound organelle found in eukaryotic cells. It serves as the cell's control center, housing genetic material and directing cellular activities. Structure. The nucleus is typically spherical and one of the largest organelles in the cell. It is enveloped by a double-layered nuclear membrane, nuclear envelope, with nuclear pores that regulate the passage of molecules. Genetic material. The nucleus contains the cell's genetic material, which is organized into chromosomes. Chromosomes are composed of DNA and associated proteins. Genes within the DNA provide instructions for synthesizing proteins and regulating cellular processes. Nucleoplasm. The nucleus is filled with a semi-fluid substance called nucleoplasm. This environment supports various nuclear processes. Nucleolus. The nucleolus is a distinct region within the nucleus. It is involved in the synthesis and assembly of ribosomal RNA, NR, and ribosomes, which are essential for protein synthesis. Gene expression. The nucleus plays a central role in gene expression. It regulates the transcription of DNA into RNA, which is then processed and transported to the cytoplasm for protein synthesis. Cellular control. The nucleus controls cell activities by managing the synthesis of proteins, enzymes, and other molecules necessary for cell function. It responds to signals from the cell's environment, influencing growth, division, and adaptation. Mitosis and meiosis. The nucleus is pivotal in cell division. During mitosis, the nucleus duplicates and distributes genetic material to daughter cells. In meiosis, it enables the formation of gametes, sex cells. DNA replication. DNA replication, crucial for cell division and genetic continuity, occurs in the nucleus. The DNA unwinds, and complementary nucleotides are added, resulting in two identical DNA molecules. Transcription and RNA processing. Transcription, the process of copying DNA into RNA, occurs within the nucleus. RNA molecules are then modified and processed before exiting to the cytoplasm for translation. Regulation and differentiation. The nucleus contributes to cell specialization, differentiation, by controlling which genes are expressed in a specific cell type. Interaction with cytoplasm. The nucleus communicates with the cytoplasm through nuclear pores, allowing molecules to move in and out. Messenger RNA, mRNA, produced in the nucleus carries genetic information to the cytoplasm for protein synthesis. Health and disease. Abnormalities in nuclear structure or genetic material can lead to various diseases, including genetic disorders and cancers. Evolutionary perspective. The nucleus distinguishes eukaryotic cells from prokaryotic cells, emphasizing its importance in the complexity of eukaryotic life. In summary, the nucleus is a pivotal organelle that manages the cell's genetic information, directs cellular activities, and regulates gene expression. It is essential for maintaining the integrity, growth, and proper functioning of eukaryotic cells. Cytoplasm. Definition. The cytoplasm is the gel-like substance that fills the interior of a cell between the cell membrane and the nucleus. It houses various organelles and is a site for numerous cellular processes. Composition. The cytoplasm consists of water, icons, organic molecules, and suspended organelles. It provides a medium for biochemical reactions to occur. Organelles and structures. The cytoplasm hosts various organelles, including the endoplasmic reticulum, Golgi apparatus, mitochondria, ribosomes, and more. These organelles perform specialized functions crucial for the cell's survival. Metabolism and biochemical reactions. Many cellular metabolic reactions take place within the cytoplasm, such as glycolysis, glucose breakdown, and protein synthesis. Enzymes present in the cytoplasm catalyze these reactions. Cellular transport. The cytoplasm plays a role in intracellular transport of materials. It allows molecules to move between organelles and provides pathways for communication. Cytoskeleton. The cytoplasm contains a dynamic network of protein filaments known as the cytoskeleton. The cytoskeleton provides structural support, shape, and helps in cell movement and intracellular transport. Protein synthesis. Ribosomes, which are either free-floating or attached to the endoplasmic reticulum, facilitate protein synthesis in the cytoplasm. Storage and secretion. The cytoplasm may serve as a storage site for nutrients, icons, and other cellular materials. Cells can store substances like glycogen or lipids in the cytoplasm. Energy production. Mitochondria, present in the cytoplasm, are responsible for cellular respiration, generating energy in the form of ATP. Cell division. During cell division, the cytoplasm is distributed between daughter cells. Cytokinesis, the final step of cell division, involves the division of cytoplasm to create two separate cells. Chemical reactions. Various chemical reactions, including synthesis and degradation of molecules, occur within the cytoplasm. Protection and cushioning. The cytoplasm surrounds and protects organelles from physical damage. It also acts as a cushion, absorbing shocks and impacts. Cellular response to stimuli. The cytoplasm is involved in signal transduction, relaying external signals to organelles to initiate cellular responses. Role in homeostasis. The cytoplasm helps maintain cell stability by regulating the concentrations of icons, molecules, and water. Diverse functions. The cytoplasm's versatility allows it to participate in a wide range of cellular processes, making it essential for cell survival and function. In summary, the cytoplasm is a dynamic and multifunctional region within the cell where various organelles, processes, and biochemical reactions occur. It contributes to the cell's structure, metabolism, transport, and overall functionality. Eukaryotic cells. Nucleus. Eukaryotic cells have a distinct nucleus enclosed by a nuclear envelope. The nucleus houses genetic material, DNA, organized into chromosomes. Membrane-bound organelles. Eukaryotic cells contain membrane-bound organelles, such as mitochondria, endoplasmic reticulum, Golgi apparatus, lysosomes, and more. These organelles perform specialized functions within the cell. Size. Eukaryotic cells are generally larger and more complex than prokaryotic cells. Cell division. Eukaryotic cells divide through a process called mitosis, ensuring equal distribution of genetic material to daughter cells. Sexual reproduction involves meiosis and the formation of gametes. Genetic material. DNA in eukaryotic cells is linear and complex with proteins, histones. It is found within the nucleus and organized into multiple chromosomes. Examples. Examples of eukaryotic cells include cells in plants, animals, fungi, and protists. Prokaryotic cells. Nucleus absence. Prokaryotic cells lack a true nucleus. Instead, their genetic material is found in the nucleoid region, an area without a membrane. Lack of membrane-bound organelles. Prokaryotic cells do not have membrane-bound organelles like mitochondria or endoplasmic reticulum. Size. Prokaryotic cells are generally smaller and simpler compared to eukaryotic cells. Cell division. Prokaryotic cells divide through a process called binary fission, which results in two genetically identical daughter cells. Genetic material. DNA in prokaryotic cells is circular and lacks histones. It floats freely within the nucleoid region. Examples. Bacteria and archaea are examples of prokaryotic cells. Key differences between eukaryotic cells and prokaryotic cells. Nucleus presence. Eukaryotic cells have a true nucleus, while prokaryotic cells lack a nucleus. Organelles. Eukaryotic cells contain membrane-bound organelles, prokaryotic cells lack such organelles. Genetic material. Eukaryotic cell DNA is linear, complexed with histones, and found in the nucleus. Prokaryotic cell DNA is circular and located in the nucleoid region. Size and complexity. Eukaryotic cells are larger and more structurally complex. Prokaryotic cells are smaller and simpler. Cell division. Eukaryotic cells divide through mitosis and meiosis. Prokaryotic cells divide through binary fission. Examples. Eukaryotic cells include plant, animal, fungi, and protist cells. Prokaryotic cells include bacterial and archaeal cells. These differences in cellular structure and organization highlight the distinction between eukaryotic and prokaryotic cells, which represent two major categories of life forms. Cytoplasm. Definition. The cytoplasm is a gel-like, semi-fluid substance that fills the interior of a cell between the cell membrane and the nucleus in eukaryotic cells. Composition. The cytoplasm is primarily composed of water, various molecules, and organelles suspended within it. Organelle support. It provides structural support to the cell organelles, allowing them to maintain their positions and carry out their functions effectively. Metabolic activities. Many essential cellular metabolic reactions occur within the cytoplasm, such as glycolysis, glucose breakdown, protein synthesis, and various enzyme-mediated processes. Cellular transport. The cytoplasm serves as the medium for intracellular transport, allowing molecules and substances to move within the cell. Enzymatic reactions. Various enzymes present in the cytoplasm catalyze biochemical reactions necessary for the cell's functioning. Storage and metabolism. Nutrients, ions, waste products, and other cellular materials can be stored and processed in the cytoplasm. Cellular movement. Components like the cytoskeleton, which includes microtubules and microfilaments, are present in the cytoplasm. They enable cellular movement, shape maintenance, and intracellular transport. Ribosome function. Ribosomes, essential for protein synthesis, can be found free in the cytoplasm or bound to the endoplasmic reticulum. Energy production. Mitochondria, responsible for energy production through cellular respiration, are located in the cytoplasm. Endocytosis and exocytosis. Processes like endocytosis, cellular intake of substances, and exocytosis, release of substances outside the cell, involve interactions with the cytoplasm. Response to stimuli. Cellular responses to external stimuli, such as signals from the environment or other cells, often involve reactions within the cytoplasm. Water and ion balance. The cytoplasm's water content and ion concentration are vital for maintaining osmotic balance and proper cellular functioning. Viscosity and fluidity. The cytoplasm's viscosity and fluidity influence cellular movement and the diffusion of molecules. Cell differentiation. During cell differentiation, changes in the cytoplasm contribute to the specialization of cells for specific functions. In summary, the cytoplasm is a dynamic, versatile substance that houses organelles, facilitates metabolic activities, and supports various essential cellular processes. It plays a central role in maintaining the overall functionality and structure of the cell. Cell organelles. Cell organelles are specialized structures within cells that perform specific functions, contributing to the overall organization, growth, and functioning of the cell. Each organelle has a distinct role, and their collective efforts enable cells to carry out essential processes for life. Some key organelles include 1. Nucleus, contains genetic material, DNA, and controls cell activities. 2. Mitochondria, produces energy through cellular respiration. 3. Endoplasmic reticulum, involved in protein and lipid synthesis. 4. Golgi apparatus, modifies, sorts, and packages molecules for transport. 5. Lysosomes, digests waste materials and cellular debris. 6. Plastids. Plastids are present only in plant cells. 7. Vacuoles, stores materials like water, nutrients, and waste products. 8. Cell membrane controls entry and exit of substances, maintains cell structure. 9. Cytoplasm, gel-like substance that houses organelles and facilitates cellular processes. Nucleus. The nucleus is a membrane-bound organelle found in eukaryotic cells. It serves as the control center of the cell and contains the genetic information necessary for cell function and reproduction. The nucleus has several important functions. Genetic storage. The nucleus houses the cell's DNA, which carries the instructions for building and maintaining the entire organism. DNA is organized into chromosomes, and each chromosome contains multiple genes that code for specific proteins and traits. Gene expression. The nucleus is involved in gene expression, which is the process by which the information encoded in DNA is used to synthesize proteins and other molecules. This process involves transcription, copying DNA into RNA, and translation, building proteins based on the RNA code. Cellular regulation. The nucleus controls the overall activities of the cell. It regulates which genes are active at any given time, allowing the cell to respond to various internal and external signals and adapt to changing conditions. Cell reproduction. During cell division, the nucleus plays a crucial role. It ensures that each daughter cell receives a complete and accurate copy of the genetic information. This process is essential for the growth, development, and repair of tissues in multicellular organisms. Nucleolus formation. Within the nucleus, there is a region called the nucleolus, which is involved in producing ribosomes. Ribosomes are essential for protein synthesis, and the nucleolus helps assemble the components needed for ribosome production. In summary, the nucleus is the command center of the cell, housing and regulating the genetic information required for various cellular processes, growth, and reproduction. Mitochondria. Mitochondria are double-membraned organelles found in the cells of most eukaryotic organisms. They are often referred to as the powerhouses of the cell due to their primary role in producing energy in the form of adenosine triphosphate, ATP, through a process called cellular respiration. Mitochondria have several important functions. ATP production. Mitochondria carry out aerobic respiration, a complex biochemical process that converts glucose and oxygen into ATP, the main energy currency of the cell. This energy-rich molecule is used to power various cellular activities, including muscle contraction, active transport, and chemical reactions. Krebs cycle, citric acid cycle. This cycle, which occurs within the mitochondria's inner matrix, breaks down the products of digestion, generating energy-rich molecules like NAD and FADH2. These molecules then contribute to the electron transport chain, the final step in ATP production. Electron transport chain, found in the inner mitochondrial membrane. This chain consists of a series of protein complexes that transfer electrons. As electrons move along the chain, they release energy, which is used to pump protons, H+ ions, across the inner mitochondrial membrane, creating an electrochemical gradient. ATP synthesis. The electrochemical gradient established by the electron transport chain drives the enzyme ATP synthase, which generates ATP from adenosine diphosphate, ADP, and inorganic phosphate Pi. Calcium regulation. Mitochondria also play a role in maintaining cellular calcium levels. They can sequester calcium ions, which are important for signaling and cell function. Proper calcium regulation is essential for processes like muscle contraction and cell communication. Apoptosis regulation. Mitochondria are involved in programmed cell death, or apoptosis. They release certain molecules that trigger apoptosis when the cell is damaged or no longer needed. This is a crucial process for maintaining proper tissue development and removing damaged cells. In summary, mitochondria are essential organelles that generate energy for the cell through the production of ATP. Their functions extend beyond energy production and include roles in regulating calcium levels and participating in programmed cell death. Endoplasmic reticulum. The endoplasmic reticulum, is a complex organelle composed of a network of membranes within eukaryotic cells. It plays a crucial role in various cellular processes, particularly protein and lipid synthesis. There are two types of rough smooth each with distinct functions. Rough endoplasmic reticulum, rough or studded with ribosomes on its surface, giving it a rough appearance. Main function is protein synthesis and modification. Ribosomes on the rough or synthesize proteins that are destined for secretion, incorporation into the cell membrane, or transport to other organelles. As proteins are synthesized, they enter the rough ER's lumen, anterior space, for initial folding and modifications, such as glycosylation, attachment of sugar molecules. Smooth endoplasmic reticulum, smooth lacks ribosomes and appears smooth. Primarily involved in lipid metabolism and detoxification. Synthesizes lipids, including phospholipids and steroids, which are important components of cell membranes and cellular structures. Participates in detoxification processes by metabolizing drugs, toxins, and other harmful substances. Stores and regulates calcium ions, which are important for various cellular processes including muscle contraction and cell signaling. Overall, the endoplasmic reticulum plays a central role in the synthesis, modification, and transport of proteins and lipids within the cell. It contributes to the overall structure of the cell membrane and is involved in various essential cellular functions, making it a critical organelle for cell survival and proper functioning. Golgi apparatus. The Golgi apparatus, often referred to as the Golgi complex or Golgi body, is a membrane-bound organelle found in eukaryotic cells. It plays a key role in the processing, modification, sorting, and packaging of proteins and lipids before they are transported to their final destinations within the cell or outside of it. The Golgi apparatus consists of flattened membrane sacs called cisternae, and its functions include protein modification, proteins that are synthesized in the endoplasmic reticulum, and enter the Golgi apparatus undergo various modifications. These modifications can include the addition of carbohydrate groups, glycosylation, which can affect the protein stability, function, and targeting. Protein sorting. The Golgi apparatus sorts proteins based on their destinations. It can direct proteins to various parts of the cell, such as lysosomes, the cell membrane, or secretion outside the cell. This sorting is facilitated by specific molecular markers added during the Golgi processing. Formation of lysosomes and secretory vesicles. The Golgi apparatus is responsible for forming lysosomes, which are organelles containing enzymes that break down cellular waste and macromolecules. It also forms secretory vesicles that carry modified and packaged proteins to the cell surface for release through exocytosis. Lipid metabolism. In addition to processing proteins, the Golgi apparatus is involved in lipid metabolism. It synthesizes certain lipids, modifies others, and packages lipids into vesicles for transport. Formation of cell wall. In plant cells, the Golgi apparatus is involved in the synthesis and modification of components that contribute to the cell wall structure, including cellulose and other polysaccharides. Membrane renewal. The Golgi apparatus contributes to the renewal and maintenance of cellular membranes by adding new lipids and proteins to them. In summary, the Golgi apparatus acts as a central hub for processing, modifying, sorting, and packaging cellular molecules, especially proteins and lipids. Its intricate functions are vital for ensuring that these molecules are properly modified and directed to their appropriate destinations within the cell or beyond. Lysosomes. Lysosomes are membrane-bound organelles found in eukaryotic cells, and they play a crucial role in cellular digestion, waste disposal, and recycling. They contain a variety of enzymes that are capable of breaking down different types of biomolecules. Lysosomes have several important functions. Intracellular digestion. Lysosomes contain hydrolytic enzymes that can break down complex molecules, such as proteins, nucleic acids, carbohydrates, and lipids, into their simpler building blocks. This process is known as hydrolysis, where water molecules are used to break chemical bonds. Waste management. Lysosomes are responsible for digesting and recycling cellular waste, damaged organelles, and other materials that are no longer functional. This process, called autophagy, helps maintain cellular health by removing potentially harmful components. Phagocytosis. Lysosomes are involved in phagocytosis, the process by which cells engulf and digest external particles such as bacteria, viruses, and cellular debris. The lysosomes fuse with the phagosomes, membrane-bound compartments formed during phagocytosis, to digest the ingested materials. Programmed cell death, apoptosis. Lysosomes can also play a role in programmed cell death, apoptosis. In some cases, lysosomal enzymes are released into the cytoplasm, causing the breakdown of cellular components and leading to the controlled dismantling of the cell. Bone remodeling. In specialized cells called osteoclasts, lysosomes contribute to bone remodeling by breaking down and resorbing old or damaged bone tissue. Metabolic regulation. Lysosomes are involved in regulating cellular metabolism by controlling the availability of nutrients. They release digested molecules back into the cytoplasm for reuse. Despite their powerful digestive capabilities, lysosomes are typically well contained within the cell to prevent the enzymes from damaging the surrounding cellular structures. The integrity of lysosomal membranes is crucial for maintaining cellular health. In summary, lysosomes are responsible for digesting, recycling, and managing cellular waste and play a crucial role in maintaining the overall health and functionality of eukaryotic cells. Plastids. Plastids are double-membraned organelles found in plant cells and some algae. They are involved in various essential processes, including photosynthesis, pigment synthesis, and storage. There are different types of plastids, each with specific functions. 1. Chloroplasts. Chloroplasts are the most well-known type of plastids and are responsible for photosynthesis. They contain chlorophyll, a green pigment that captures sunlight and converts it into chemical energy in the form of glucose through the process of photosynthesis. Chloroplasts have inner membranes organized into stacks called granular, where the light-dependent reactions of photosynthesis occur, and a fluid-filled region called the stroma, where the light-independent reactions, Calvin cycle, take place. 2. Chromoplasts. Chromoplasts are responsible for synthesizing and storing pigments other than chlorophyll, giving fruits and flowers their various colors. They produce pigments like carotenoids, orange, red, and yellow pigments, and provide visual cues to attract pollinators or seed dispersers. 3. Amyloplasts. Amyloplasts are plastids that store starch, a complex carbohydrate formed through photosynthesis. They are often found in non-photosynthetic tissues like roots, tubers, and seeds, where they store energy in the form of starch for later use by the plant. 4. Leucoplasts. Leucoplasts are a general category of plastids that can develop into various specialized types based on the presence of different enzymes and pigments. Some leucoplasts are involved in synthesizing fatty acids, lipids, and other molecules required for cellular functions. Plastids can interconvert between different forms based on the needs of the cell in the developmental stage of the organism. For instance, chloroplasts can transform into chromoplasts during fruit ripening, changing the color of the fruit as pigments are synthesized. The transformation of plastids is regulated by the plant's genetics and environmental factors. In summary, plastids are multifunctional organelles found in plant cells, with roles ranging from photosynthesis and pigment synthesis to energy storage and the synthesis of various cellular components. Vacuoles. Vacuoles are membrane-bound organelles found in the cells of plants, fungi, and some protists. They serve various functions, primarily related to storage, waste management, and structural support. Vacuoles can vary in size and function depending on the type of cell and organism. Here are their key functions. Storage. Vacuoles store a variety of substances, including water, ions, nutrients, such as sugars, amino acids, and minerals, and waste products. These stored materials can be used by the cell when needed, such as during periods of drought or nutrient scarcity. Turgor pressure and cell rigidity. Plant cells. In plant cells, the central vacuole plays a crucial role in maintaining turgor pressure. By storing water and ions, the vacuole helps create internal pressure that maintains cell shape and rigidity. This pressure contributes to the overall structural integrity of the plant. Waste management. Vacuoles are involved in breaking down and storing cellular waste products. This function is particularly important in fungi and plant cells, where vacuoles help detoxify harmful compounds and store waste until they can be safely removed. Pigment storage. Some vacuoles store pigments that contribute to the coloration of flowers, fruits, and other plant parts. These pigments attract pollinators and seed dispersers, and they can change the appearance of plants as they develop. Defense mechanisms. In certain plant cells, vacuoles can store toxic compounds, such as alkaloids, as a defense against herbivores and pathogens. When the plant is damaged, these compounds can be released to deter or poison attackers. Digestive functions. Protists. In some single-celled organisms like protists, vacuoles are involved in intracellular digestion. They engulf food particles through a process called phagocytosis and then fuse with lysosomes to digest the ingested material. Cell growth and development. Vacuoles play a role in regulating cell growth and development. They can store molecules that are required for cell expansion and division. Vacuoles are highly dynamic organelles that can change in size and content based on the cell's needs. Their functions contribute to maintaining cellular homeostasis, supporting growth, and aiding in adaptation to changing environmental conditions. Cell membrane. The cell membrane, also known as the plasma membrane, is a crucial structure found in all types of cells. It separates the interior of the cell from the external environment and plays a variety of important functions. Cellular barrier. The primary function of the cell membrane is to act as a barrier that separates the internal contents of the cell from the surrounding environment. It controls the movement of substances into and out of the cell, allowing for selective exchange of molecules. Selective permeability. The cell membrane is selectively permeable, meaning it regulates the passage of substances based on their size, charge, and other properties. It allows essential molecules like nutrients and gases to enter the cell while keeping unwanted substances out. Transport. The cell membrane facilitates the transport of ions, nutrients, and other molecules across its lipid bilayer. This can occur through various mechanisms, such as passive diffusion, facilitated diffusion, active transport, and bulk transport, endocytosis and exocytosis. Cell signaling. The cell membrane contains proteins that serve as receptors for various signaling molecules, such as hormones and neurotransmitters. When these molecules bind to specific receptors on the membrane, they initiate intracellular signaling pathways that regulate cellular activities. Cell adhesion. The cell membrane plays a role in cell adhesion, allowing cells to stick to each other and form tissues. Proteins on the cell surface called adhesion molecules help cells interact with neighboring cells and the extracellular matrix. Cell recognition. The cell membrane carries glycoproteins and glycolipids on its surface that are involved in cell recognition. These molecules help cells identify and communicate with each other, participate in immune responses, and distinguish self from non-self. Fluidity in structure. The lipid bilayer of the cell membrane provides flexibility in fluidity to the membrane, allowing it to change shape and accommodate various cellular processes. Cholesterol molecules within the membrane help maintain its stability and prevent excessive fluidity. Protection and defense. The cell membrane serves as a protective barrier against harmful substances and pathogens. It prevents the entry of potentially damaging molecules and microbes, helping to maintain the cell's internal environment. In summary, the cell membrane is a vital component of cells that performs essential functions related to maintaining cellular integrity, regulating transport, facilitating communication, and interacting with the external environment. It is a dynamic and adaptable structure critical for the survival and proper functioning of cells. Cytoplasm. Cytoplasm is the gel-like substance that fills the interior of a cell, surrounding organelles and the nucleus. It plays several important roles in supporting cellular processes and maintaining cell structure. Cellular metabolism. Many biochemical reactions take place within the cytoplasm. It serves as a medium where various metabolic pathways occur, including glycolysis, breakdown of glucose, protein synthesis, and lipid metabolism. Enzyme activity. Enzymes, which are critical for catalyzing biochemical reactions, are often present in the cytoplasm. They help drive metabolic processes necessary for cell survival and function. Cellular transport. the cytoplasm contains ions, molecules, and nutrients required for cellular activities. It serves as a medium for transporting these substances within the cell through processes like diffusion, active transport, and endocytosis, exocytosis. Support and structure. The cytoplasm provides structural support to organelles and the cell's internal components. It helps maintain the cell's shape and acts as a scaffold for cellular organization. Cellular communication. Some signaling molecules and proteins involved in cellular communication and signaling are present in the cytoplasm. These molecules can affect cellular responses and gene expression. Storage. The cytoplasm can store various molecules, including nutrients and ions, for later use by the cell. It plays a role in storing glycogen in animal cells and starch in plant cells as a source of energy. Cytoskeleton organization. The cytoskeleton, a network of protein filaments and tubules, is found in the cytoplasm. It provides structural support, helps maintain cell shape, and is involved in cell movement, division, and transport of organelles. Cell division. During cell division, the cytoplasm is distributed between daughter cells. It contains the necessary components for both mitosis, division of the nucleus, and cytokinesis, division of the cytoplasm. Protein synthesis. Ribosomes, which are responsible for protein synthesis, can be found floating freely in the cytoplasm. They read the instructions encoded in mRNA and synthesize proteins accordingly. In summary, the cytoplasm is a dynamic and multifunctional substance within cells that supports various cellular activities, ranging from metabolism and transport to structure and cell division. It provides a medium for biochemical reactions, maintains cell integrity, and plays a central role in cellular function and homeostasis. Cell division. Cell division is a fundamental process by which cells reproduce and contribute to growth, development, and tissue repair. It involves several stages and mechanisms to ensure the accurate distribution of genetic material and organelles between daughter cells. Here's an overview of cell division. Purpose. Cell division allows organisms to grow, replace damaged cells, and produce new cells for reproduction. Types. There are two main types of cell division, mitosis and meiosis. Mitosis produces identical daughter cells for growth and repair. Meiosis generates gametes, sperm and egg cells, with half the chromosome number for sexual reproduction. Mitosis. Mitosis is a process of somatic, body, cell division. It consists of stages: interphase, prophase, metaphase, anaphase, and telophase. Interphase. The cell prepares for division by growing, replicating DNA, and synthesizing organelles. DNA is in chromatin form during this phase. Prophase. Chromatin condenses into visible chromosomes. The nuclear envelope disassembles, and spindle fibers begin to form. Metaphase. Chromosomes align at the cell's equatorial plane, metaphase plate. Spindle fibers attach to centromeres of each chromosome. Anaphase. Sister chromatids separate and move towards opposite poles of the cell. The cell elongates as the spindle fibers shorten. Telophase. Chromatids reach the poles and decondense into chromatin. The nuclear envelope reforms around each set of chromosomes. Cytokinesis. After mitosis, cytokinesis occurs, dividing the cytoplasm and organelles into two daughter cells. In animal cells, a contractile ring of actin filaments pinches the cell membrane. In plant cells, a cell plate forms, eventually developing into a new cell wall. Meiosis. Meiosis occurs in specialized cells to produce gametes with half the chromosome number. It consists of two rounds of division, meiosis I and meiosis II. Meiosis I. Homologous chromosomes, pairs, separate, reducing the chromosome number by half. This generates two haploid cells with unique combinations of genetic material. Meiosis II. Similar to mitosis, sister chromatids separate in each haploid cell. 4 haploid daughter cells are produced at the end of meiosis II. Genetic diversity. Meiosis leads to genetic diversity due to the random assortment of chromosomes and genetic recombination, crossing over, during prophase I. Regulation. Cell division is tightly regulated by checkpoints to ensure accurate DNA replication and chromosome segregation. Failures in regulation can lead to genetic mutations and diseases like cancer. Significance. Cell division is vital for growth, development, tissue repair, and reproduction in multicellular organisms. It contributes to the maintenance of a stable chromosome number across generations. In summary, cell division is a complex and carefully orchestrated process that ensures the proper distribution of genetic material and cellular components between daughter cells. It plays a central role in the continuity of life and the maintenance of genetic diversity within a species. Thank you for watching. Please like, share and subscribe to my channel LinkedIn Farmer.